So welcome to the brief presentation of dBrain. It stands for Digitizing Brain Data for Health and Disease. So I'm Jeanette Hellgren-Kotaleski, the formal coordinator of dBrain, but in reality, a lot of faculty are involved in this, including their postdocs and students. dBrain is a multidisciplinary collaboration between three KTH environments: math, computational science, and health science. And we also have collaborators at the Karolinska Institute and Karolinska Hospital, and they provide a lot of data, but also obviously a lot of expertise regarding the clinics. So the motivation behind dBrain is that we have to conquer brain disorders because it's already covering more than 35% of the healthcare cost in the Western world, and this is increasing, not to speak about all the suffering patients and relatives have to endure. But fortunately, we are also living in the era of big data. So thanks to various technological developments during the last few decades, we can measure, especially in animal models on different scales, what happens in the brain in health and disease, but we can also do a lot of measurements in human patients. And then the challenge is to go from data to new insights and knowledge and understanding, and this we can do through modeling and simulations and through the use of AI and machine learning. So the typical situation today is that when you get a brain disorder and become a patient, you go to the doctor and describe your symptoms, and the doctor will then take some tests or biomarkers to validate the diagnosis, and then they set up a treatment plan. And I guess that's fine, but the reality is that many brain disorders are very hard to treat, and you might just treat symptoms, but not the causes of the disease. And this is a challenge, and dBrain try to address this with adding on this blue box, where we try to use all available data, and we use AI-based data analysis methods. And with this, we will find correlations between biomarkers and the disease or the disease progress, or predict how well a particular treatment will work and so on. If we want to understand also the causal chain of events or the mechanisms behind the disease symptoms, we also, and development of the disease, we also need to use computational models of the brain. This means that we already in the model represent what is known, and then we use data to constrain the model parameters, and we can map model variables onto biological entities. So these things are done in parallel, and in this project we focus on motor and neurodegenerative diseases such as Parkinson's disease and Alzheimer's disease. So in dBrain we use a three-way attack on brain diseases. At the finest scale, we look at single neurons and try to predict network behavior based on that. And at mesoscale level, we might use data relating to fMRI or EEG data. And at the behavioral level, we can also analyze motor performance. So I will now start with exemplifying this cellular level scale. So in Parkinson's disease, it's known that neurons degenerate, especially in a particular part of the brain called the stratum. So in the stratum, we have two main cell types called SPNs that degenerate. So this is a sort of control case, and during different stages of Parkinson's disease progress, they start to lose their distal dendrites, so they shrink. But at the same time, interneurons in the same nucleus of the brain, they could even grow axons. They don't shrink, but they grow axons instead. And we want to understand then how these cellular level changes, what consequences it has for network properties when it comes to structure as well as dynamics. So we have already set up a workflow for the healthy brain, and now we apply it to the diseased brain. So the workflow starts with that we distribute neurons with the right density in the volume we want to simulate. And then we predict synaptic connectivity. This is the red dots between neurons by looking at where axons and dendrites come close. And then we apply pruning rules based on what is known about how neurons connect in the brain. This is just an example where we use one neuron of that type, one neuron of that type, and one neuron of that type. This is a control case. And the red dots are where these neurons connect to each other. In the Parkinsonian case, these neurons lose part of their dendrites, and then obviously the corresponding synapses disappear, but these interneurons might add on new synaptic contacts instead. And we can then in the network measure things like what is the likelihood that two neurons with a certain distance between them talk to each other. But we can also do it in a more quantitative and more elaborate way by looking at something called clicks, which is all-to-all connected nodes. This is an example of a click of order 2. So we look at one neuron and how it connects to another neuron, and it can do that directly, but it can also do it via another neuron. So this is another example with a click of higher dimension. This has a dimension 5. And then when we analyze the network during progressive stages of Parkinson's disease, we see that this is an example of the healthy network, but when Parkinson's disease changes progress, the richness of these motives decreases significantly. And it's interesting that if we ablate interneurons, this is decrease of the richness of these clicks decreases very rapidly. But that is not actually what happens in the control network, which is very robust to changes in interneurons, for instance. We can also look at what type of neurons are in these different clicks, the composition of neurons, and not unexpectedly, if we have at least one interneuron in the clicks, they are much more robust to degradation due to Parkinson's disease. So this is an example of how we can go from cellular level to network and look at structure. We also simulate this. I don't have time to talk about this, but we really believe that to disentangle structural and dynamic mechanisms in the network could lead to novel targets for drug discovery in the future. So that was an example of the detailed level, and now we go to the mesoscale level.
Hi, my name is Arvind Kumar. I'm one of the PIs in the dBrain project. I will briefly update you on the development of mesoscale models of Parkinson's disease and how we are using those models to not only get insights into the disease mechanisms, but also to develop a model-driven data analysis pipeline to analyze the brain activity and to improve the diagnosis of the disease. One of the key signatures of Parkinson's disease is the so-called beta band oscillations. These oscillations are about 20 Hertz in frequency, and these oscillations appear when patients show motor symptoms of the disease and disappear, for instance, when they switch on the therapeutic dBrain stimulation. We have understood that the cause of these oscillations is somewhere in the basal ganglia, the key structure which is impaired in the disease. It is not really clear among all these different loops in the basal ganglia which of the loops is responsible for these oscillations. Experimentalists are desperately trying to find out the target loops. To better understand this, we actually turn to mathematics, and first of all, we developed a formal proof to understand what are the structural constraints that give rise to these kind of oscillations. And our proof could be summarized in a very simple rule, which is that take a cycle and count the number of inhibitory connections in that. If the number of inhibitory connections is odd, then the network can oscillate, otherwise it won't. So this gives us a very nice insight into the basal ganglia that perhaps it's not so advisable to search for a specific cause of these oscillations. There are many possible sources of these oscillations, and if one is not working, other may take over. Irrespective, this analysis led to the understanding that oscillations indeed entail an imbalance of excitation and inhibition, and also that if you want to properly place the electrode, one should consider the degree of imbalance between excitation and inhibition. And this is a quite new insight that we think that clinicians would be very much interested in, as they often struggle in placing the DBS electrodes. These results have been accepted for publication in eLife and will soon appear. Based on these analyses, we ventured out to understand whether the disease-related changes are only localized in the basal ganglia or they spread out across the whole brain. And for that, we made a collaboration with Nutmeg facility in Sweden. They provided us data recorded from patients and control subjects. The data came in the form of magnetoencephalogram, and it looks something like this. It's just a time series corresponding to different locations in the brain. The key idea here is that came from our models is that instead of just looking at classical peaks in this spectrum of the activity, like what kind of oscillations are there, our analysis suggested that we should be instead looking into the aperiodic activity, that is the slope of the spectrum, how it decays as a function of frequency. And this is something we quantified with lambda. And the reason we are interested in this parameter is that it is again related to the excitation and inhibition balance that I previously talked about. When we compare the lambda measured from healthy subjects and Parkinson patients without medication, we find that there are massive changes across the neocortex, something that has never been reported before. Specifically, we found that sensory areas, which are usually not the target of clinical investigations in PD, there we find a major increase in the exponent, which translates according to our model into the fact that in the sensory areas, there is an excessive inhibition. Now, surprisingly, this increase in inhibition is not affected when patients took medication. So current medication and the diagnostic batteries, they don't take into account of the sensory deficits. So this is quite a new finding, and it basically suggests that first we should consider sensory deficits as a key diagnostics variable. And if that comes out, we will have an opportunity to relate this particular measure to the degree of PD severity. And it also asks many other questions that we are addressing currently. For instance, what's the origin of these things? Why the lambda is affected? Is it local? Is it global? Meaning long-range interactions. But also we are interested in finding out whether deep brain stimulation, which is a de facto most effective strategy to alleviate the symptoms, whether deep brain stimulation somehow reverses these effects, unlike the levodopa medication. So that's the progress so far in mesoscopic modeling and model-driven data analysis of Parkinson's disease. Next, we will hear about how we can use geometry to understand the dynamics of this activity in Parkinson's disease. Thank you very much.
Hello, my name is Andrea Guidolin, and I will now briefly speak about how geometry can help us to understand complex and high-dimensional data. I'm a postdoc at KTH in the math department in the group of Wojciech Hachulski, and the focus of our research is on topological and geometric methods for data analysis. In dBrain, we provide support on the use of such methods, which are well suited for applications to neuroscience and in general to complex biological data. In data analysis, geometry is used in many common methods, such as PTA or clustering, to extract information from the data. To use geometric methods in data analysis, one has to first consider some spatial representation of the data, such as a graph or a network, or a sequence of them, or a high-dimensional generalization of them. The role of this spatial representation of the data is not simplifying and directly providing information of the data, as the complexity of the spatial representation is usually the same as the original data. The advantage of having such a spatial representation of the data is that one can extract some geometric descriptors, also called invariants, which can provide insights on the data. An example of such descriptors are some counting statistics or some geometric motifs, such as the click count that has been used to characterize brain networks associated to different stages of Parkinson's disease, as Jeanette presented before. Another invariant that is quite useful and powerful to understand complex data is homology.
Hello, my name is Rodrigo Moreno. I'm working in Orient Smith group. We have been working with optimized structural connectivity. We developed different advanced tractogram filtering methods to increase the reliability of structural connectivity analysis. So we have our methods to process diffusion MRI data, and then with that we can classify patients and controls. We have been working with both Parkinson's patients and Alzheimer's patients using this methodology in these papers. We have been also working with synthetic MRI aging. So we are able to create synthetic MRI images at different ages. And we can also create templates of healthy brains at different ages. And we use them to see the evolution of the Alzheimer's disease progression compared to the healthy trajectory. We can see here in this image the quality of the synthetic images that we are generating. So they are very similar to those that are acquired for the real patient.
Hello, my name is Gonzalo Uribarri. I'm a postdoc working with Eric Francien at KTH University. And I will talk about the behavior part of dBrain. In one of our projects, we are working alongside with medical doctors at Karolinska Hospital, where they have patients with tremors. We have developed an app that can measure the shaking of the muscles and allows us to monitor and characterize the tremor using signal analysis. The goal is to have a measurement that can assist in diagnosis and quantify the effect of medication. In other of our projects, we are working with Per Spegningsson at Karolinska Institute, where they have patients with Parkinson's disease. In these projects, we analyze eye movement data from eye tracking experiments using machine learning. The goal is to find biomarkers that can differentiate patients from healthy controls. These eye movement biomarkers are a non-invasive measurement that can help in diagnosis and prognosis of the disease. This was a short introduction to dBrain. If you want more information, you can visit our website, and thank you for listening.